Okay, let's get it started. So this week we are going to touch base on another very important topic in order to establish your machine learning understanding thoroughly. So last couple of weeks we majorly go through linear algebra. So we play with vectors, matrix, how to do the multiplication of matrices, and also use the squared matrix as a rotator or operator to change other vectors' direction or magnitude. So this week, from my understanding, so the machine learning is built on these two pillars. One is linear algebra, another will be this probability theory and statistics for sure. And the rest of them are important but not necessary. So if you don't know calculus, you don't know how to calculate those derivatives, it's okay. You can still using this optimization black box to do this optimization. So you don't even know what's going on inside, but it still works. But you still have to know the basic terminology in linear algebra. You know the eigenvalue, eigenvectors, and then a probability distribution, what that means, especially today, is very important. Everybody heard about the generative model? Generative models, yeah. This is hot topic recently, but then, okay, generative model. So model is a statistical models, and so today we have to solve the problem. And what is a model? When statisticians talk about modeling and build a model, what that means, and also we will say, okay, once you understand what the probability is, what's the distribution, what the generative means. Generative basically just I will generate new data points which wasn't there, but based on some of the understanding of your model. So generative model can be used, for example, ChatGPT is a large language model. It is a trained generative model. The ChatGPT, the model, what it does is to generate the next word based on the probability. If 95% should be this word, the generator will pop up this word 95%. And then next. Just more complex than the one we are going to discuss. Like the simplest model is, what do you think the simplest model should be? I have a distribution like this. So in this axis is your value, and a number for your data. Okay, we generate a series number, and this side is the frequency. Okay, and then if you have a continuous variable, this value will be not discrete. So, but we can prepare this so-called distribution or function. Probability distribution PDF function. By bin them, we can bin them. Say from this value to here, what's the frequency, and what's that frequency? Basically, you build a histogram, okay? If it's a continuous variable, you can make this bin very thin and just very small steps, and even you cannot see them. Maybe it's even over the precision of the computer can handle. In that case, you build a continuous variable. This is called a distribution. We are using function density, probability density function to replace the histogram. Okay, everybody knows how to do the histogram. This could be frequency. If you have numbers, you can count. It's the same. All right, you normalize it. as I said, what is the simplest one? The simplest one will be a distribution which is purely random. It will like this. My value is from 0 to 1. And my frequency for the spins are all the same. So to me, this is the simplest model I can have. What that means is if I do a generator, I will generate a model, and I will generate a series of points. But this series of points will be drawn randomly from this distribution, and because all the bars are the same height, they are equal frequent. So I can draw randomly at any point here. Okay, so if I use this as my model to generate my data, my generator is a random generator. In MATLAB, it's easy. It's a function called RAND. Okay, if you do nothing, it will give you a random number, and either located in this bin or that bin, and it generates a continuous variable. So you see a decimal number. Okay, you can run this many times, and the number will be jumping around in here. No, this distribution, the simplest distribution, generates numbers between 0 and 1. Yeah. Okay, certainly today we are going to talk about more different type of the probabilities and different type of the distributions, which more complex than this uniformly random, okay, distributed distribution. And the reason is our data, what we will see, we will observe. Many of them not follow this pattern. Okay, so another extension will be the number we got is a distribution like a Gaussian, and then in that case, you centered at 0, and then you histogram looks like this, and make it continuous, and then it become a normal distributed curve. Okay. In that case, we run random generator. If based on this, we will generate, for example, negative 0.52 and 0.3 positive. So we will generate data points which centered around 0, but there possibility you can extend your value to this end or that end. Okay. So you can do this by not generate random n, but the function is called RAND. N means normal distributed. So in this case, the difference, I don't know if you can see it clearly. This function generates uniformly distributed random number between 0 and 1. And this RANDN generate a normal distributed random number centered around 0. And you may not see the difference. You say, okay, I generated this random number 0.6604. Seems like it's same as R&D and generated, right? You don't see the major difference. But once you run a lot, you start to see the difference, okay? So you need go back to collect your data, and you not the data may be randomly generated, but you have to collect them and do statistics on them to figure out the original generative model. Okay, we are doing the generator, using generator generate a lot of data. In other way around, you collect those data, you want to study them to infer those parameters to understand what's under the underlying distribution the generator is using. So we are doing this generative model, generate data, and using generated data to figure out what the distribution the generator is using. Make sense? Okay. So this function is different from R and D. Because they're based on the different models. Okay? So we will introduce a couple of the distribution. There are many, and this lecture is not designed for that. I can go through several of them that will be used in our single-cell data modeling. Okay, we will talk definitely and normal, Poisson, negative binomial, and the T distribution. Okay, we will describe once you have data points, you have observation, you have a lot of data, and what you can do, you can focus on the descriptive statistics. We know you can calculate mean, you can variance. There are different ways to calculate variance and to understand the variability. Then we will review again how can you compute covariance matrix, prepare for our next week's lecture in the PCA. And we also want to look at the covariance matrix and understand what that means. I will have a lot of small concepts, the terminologies. One of these is like a KL divergence. You certainly need to know this, and the people use so widely in machine learning to understand the difference between two distributions. So what's the difference? How quantify the difference between this distribution versus this or versus that? Okay, people use KL divergence to compute a number to tell you the difference between not the vector, not the data points, but the difference between distributions. Any questions? All right. So you have seen this before, and this is real data. So last computer session, we learned you can compute the summation, the column add up, and you know that it's called library size. I use a small example to see, okay, revisit library size normalization. So when you calculate the summation of the total of the rows, those are the expression total of this particular gene, and then the columns are the library size, basically how many UMIs, how many rays have been counted as a UMI in per cell, and then across all the genes, okay? And then you can see some of this have been oversampled if you consider those cells are the homogeneous population, all right? And you believe this is oversampled by some kind of bias, so you want to correct those and to introduce a factor and for those lowly expressed or sample list cells. This is normalization. We try to normalize the library size to give the results that becomes equal to all the cells by their library size. So how can we do that? We calculate a mean of the overall library size across all the cells. So on average, we have 5.29 UMI library size for the cell, and some is larger, some is smaller. Then we divide this value, okay? This factor, and then allow them, for example, this cell was 10, but after normalization, it becomes the same as another cells. So basically, you divide yourself and then multiplicate to this average across all the ones. Okay. So what do? You divide all the values by the total. Basically, you make a percentage or the portion. Okay. Then you, after divide by your total, when you sum the column again, everybody becomes 1. Okay. Then you rescaled it by the mean or some other numbers, a constant. Then everybody is the same value. So people routinely do this, and in my package we also do this, but there's a problem with that. So that's the way you do the summation, and then that's by column, and then you divide each. So this becomes library size for each cell. It's a vector. Then you use big X divide by the vector. So each column divided by this total by corresponding library size, and you normalize it into fraction, and then you multiply it by either this 10,000, or in this case is multiplied by the average. Either way. And this chart is from this figure is from this paper, and it's a paper which discussed next generation sequencing data normalization. And this is raw data. They have 1 to 3 genes. Then you have 2 samples. This is for bulk RNA-seq, but you can consider this like a gene cell 1, cell 2, the same. And if you compare these 2 samples, and you know for gene 1, the first sample is upregulated, have a high value than the control, and then the second gene, 10, 15, have a lower value than control. Well, after you do this library size normalization, the number becomes this. Okay, it still keep the same. That's what we are doing, and for this C is called RPKM. Is another way to do this normalization. This is widely used in bulk RNA-seq. Essentially, this is a library size normalization. No difference from that. The only difference is the value will be further divided by the length of a gene. Okay, so if you look at the information, there's a small number under each gene tells you how long this gene body is. If a gene is really long compared to another shorter gene, and then on average, you should expect most rates have been aligned to the longer gene. So you want to normalize those gene lengths. So that's what the RPKM is doing. After library size normalization, do one more step to normalize, to divide by the length of the gene. Okay? In that way, you can compare gene 1 with gene 2, even though they are not in the same length, gene body length. Okay, and in bulk RNA-seq world, people also use this package called DE-seq, and the DE-seq take a different strategy, and it use geometric mean in the middle, and also take this scaling factor compute for the global, and this is popular, basically like golden standard. However, if you look at the results, it changed the original, and the original look, gene 1 is higher in sample 1, and gene 2 is lower in sample 1. But like DE-seq, after doing this, and you don't see the difference between two samples in gene 2 and 3. So this is like a consequence. We don't know whether this changed the conclusion of some of these studies or distort the data and make it better or more accurate or not. So we don't know. There are many problems associated with different ways of normalization. So we do need to know how this normalization has been done in when you study working with your own data. Okay, you don't want to just follow the popular strategy. And the DE-seq is widely used in single-cell data analysis as well. A lot of people say, I want to use the DE-seq to normalize my single-cell data. We don't know the consequence yet. Okay, there's some field. We are still discussing. Yes.
What did you say the basic normalization is for DE-seq?
Yeah, DE-seq, if you look at their the package, they use geometric mean instead of average, the regular. I don't know why they use that, but that's the way they choose. Mm-hmm. So this problem even more severe because we are dealing with not different samples. So in bulk RNA-seq, people study those samples. You normally assume sample 1, sample 2, they're comparable. They have a different gene expression for particular genes, but not globally. Not to say, okay, for sample 2, all the 6,000 genes upregulated compared to sample 1. Maybe just like a different temperature, you sample those cells. But we don't assume that. But in single-cell study, you just cannot avoid those because look at this macrophage is much larger than lymphocytes and neutrophils. And so this contains more mRNAs by itself. So before you normalize it, and you assume, because your normalization is before you detect the cell type, so you still assume all the cell has the average, the similar total amount of mRNA in there. However, you see, okay, the library size for macrophage actually larger than T cells, but you still consider them as the same homogeneous population, and you try to normalize there. So you push down the library size proportionally and artificially increase the library size and make them same at the end. All the genes within these cells will be following this distortion or stretch of your rescaling of your expression. And then if you focus on one of this particular gene, it looks like gene A actually express level is higher in neutrophil than macrophage, which is not true, maybe, okay, if you know the function, maybe. So this introduced some problem, and we don't know how to solve it yet. Yeah, so people just still using library size normalization or don't do normalization at all. Yeah. One type, you still normally will do it, yeah. Mm-hmm.
But then do you need to cut off first? Let's say you cut off the extremely low one.
Yeah, so that's a QC and the quality control. You get rid of those outer layer points. But we are talking about after that step. You have this filtered and you believe the clean matrix, and then what you do next, you want to normalize, somehow normalize the library size. So to me, the best way is to do this estimation. We want to estimate each cell by given its own expected library size it should be, and maybe look at the population, different clusters, detect, for example, cluster first, and you know, okay, my population have 3 major clusters. They all have a different library size, and I will treat cluster 1, normalize it by itself, cluster 2 normalize by itself, cluster 3 by itself. But how, before you normalize it, how do you know there are 3 clusters, why not 4? So this becomes a loop. You want to learn from the data first, cluster them, and then treat them separately. So this is like a circle. You want to understand multiple parameters using this iterative learning, and to set some parameter, then learn another, then do this circle until you fit all your data nicely using some of those statistic parameters to describe your number of clusters, each cluster's mean, each cluster's average library size, each cluster's variability. In that case, you understand your data better than just treat them as a whole population. Okay. However, this procedure is computationally expensive. You have a lot of parameters to estimate and do loop again and again. So software users just not patient enough. We want to click and get it done and follow to the next step. So nobody will adapt to this in their package. This take very long time if you run this Biscuit package. Okay, but you have to know there's a better way to do it. So again, we come back to our basic concept. So I put yourself in this data analysis of a single cell RNA sequencing analysis, but this is more general. You have this function generate data, and you want to look at their distribution of and then to learn this function to understand the model. So that's your data you collected or you observed, and then you do statistics. You learn the average, the variability, and then you say, okay, my data should generate from this particular model, particular distribution, and then you can see I can draw from here to see whether I can regenerate the same kind of data or look similar artificial data sets. All right, so this is what we are doing this loop. And if you have a latest function of this tool I developed, and you will see I put a new function. So they're all the same. I reorganized the menu a little bit. So here there's a very difficult to see, but there's a data simulator. Okay, if you don't have this, you can generate a single cell data expression matrix to simulate. And I can, so I have done this, and this will ask how many cells, how many genes that you want to simulate, and then click this button, and then the procedure will be a random generator. So I will generate 0, another 0, another 15, 5 for this particular gene, and then do another. Okay, another gene maybe have a higher expression. So at the end, I generate a matrix like this. Okay, so this is the first gene and some zeros, and there are some genes maybe expression levels are higher. So if I don't tell you whether this is true or not, you may don't know whether this is from real data or not. So my example here, this one is from real data. This is from real data, and that is generated. Okay, I'm going to use this example to see how you can generate data. For example, we are trying to simulate a game that will try to generate a list of numbers. Those numbers are the height of the man in different countries. That's the European countries, and the left it will generate Italian, and they have an average 1.76 meters. And the Sweden 1.81. So these two numbers are my model. So I know they have a different average. So Italian have a lower average, where Sweden have a little bit higher. So if you want to generate this data on your mind, you will write maybe Italian, you may write 1.74, 1.75, 1.81. And then here you will automatically write a number around 1.81, 1.85, 1.79. So you know the difference, okay? So what you are doing on your mind or play this game, you are doing a generative modeling, model your data. So this is the same as what I draw on the board. If you have just a couple of data, then they are not enough. You don't see the whole pattern or whole trend of the distribution. However, once you collect enough, you can build a histogram based on how many data points in each bin, okay, the frequency. And when you collect enough, you can smooth that become a distribution, a shape. And you see the mathematical symbol changed from this summation, the sigma, become this, okay? This means continuous aggregation, okay, integrals. But in computer, and you know computer is based on the discrete 0, 1s, eventually, even you have this mathematical integral, you have to, in computer, discrete it into bins. It's just very small bins, okay? Then we have probability mass function, those for the discrete numbers, okay? And those for the density function is for the continuous. Then you have a cumulative distribution. So this is a widely used Gaussian distribution, also called a normal distribution. You have two parameters to define this curve. One is this mu, which is average, and then sigma is the standard. Standard deviation or deviation? Standard deviation. And the sigma squared is variation, okay? Variance. So can we use normal distribution to model our single cell data? So if I use normal distribution to generate my UMI count, it will look very different from what I simulated here. Remember, this is my simulation. Normal distributed value assumption will not generate this table, okay? So I have to use something different to model my single cell data. This is not my subject, but it's for statisticians, and they focus on looking at this table, what are the best distribution they should use, and what family of this distribution they should use. There are so many. Normal is here, uniform is here, okay, Poisson, negative binomial, binomial, gamma, beta, t, There are so many. What they eventually found is negative binomial distribution is the best. Okay? So we will spend 5 minutes to explain this, what it is. So when I say a binomial, I mean there's two consequences: is yes or no, or either succeed or fail. So that's binomial. And for this, the distribution is, I want to know a number, say x. So what's the probability for x? X is the number of failures until R succeeds in the independent Bernoulli trials. So in order to compute this, I need to know, okay, what's the we need to know the successful rate. Okay, we need to know P. So P, let's see whether I should use P. In that case, it's P. Small p, okay, is the probability of, say, failures, okay? If you think this like in the factory, you produce some product and the defect rate for this product is 0.05, for example. That means when you produce 100 this product, there are 5 of them may have a problem. So this is the P. So if you use a Q as a 1 minus P, so this will be the numbers of successful, okay? Let's translate this directly into single cell, okay? This number assumed to be small and this large. And in single cell, we see, okay, the P will be the mRNA will be detected. So this will be not detected. So I just translate this general term in statistics textbook and into our single cell context. So it's very difficult to detect the cell mRNA, particular mRNA in the cell. So we have a very low rate of detection, but there are high chance you will not detect it. So now we are going to compute X, for example, I detect 3 copies of particular gene. So my X is 3. I would say using negative binomial to tell me if my successful rate of detection is 0.05, I detect 3 copies of that mRNA. So how many trials I need to do before I can get a 3 success. So there's another parameters R. Is how many trials I have to do until I detect 3 copy. That's the frequency of this expected. In this small chart, there are 3 example negative binomial distribution, but using different parameter values. And the 0.6, let's look at this blue one. So 0.3 is this failure rate. And the 7 means if you do how many trials you need to do on average, for example, 15 times in order you get 7 failures. Translate into our mRNA detection is if you detect 7 copies of that gene, your detection rate is 0.3. You need on average to try 15 times. If you look at this curve, okay, most likely you have to try 15 times in order you get 7 times of that. Now I translate into mRNA detection. So you see 3 copies of your, in this case, 7 copies of your mRNA. But the real mRNA should be on average 15 or at that level on average in order to you to use 0.3% of success rate to detect 7 copies, the most likely the background should be. So there's some of this language change in order to see, okay, how many trials, so how many copies in the pool is already there. You have to use 0.3 success rate to detect 7 copies. So you can compute that. And this is the negative binomial random number generator. Okay, I showed you the uniformly random number generator and show you the normal distributed random number generator. And this is a negative binomial random generator. The table I generated is using this. Okay, but I have to set up 2 parameters for each cell and see whether how many trials you the number corresponding R. So I have to give R and P. I have to fix the detection rate, say 0.05, and I have give to this number R, which is corresponding number of success, and then to let it to generate. This is an example they used in explanation for this function. So suppose you want to simulate a process which has a defect probability. So that's a production of this factory production. At 0.01, how many units might quantity assurance inspect before found 3 defective items? So that's the way you compute using this random number, but computed 6 times. Okay, then you take average. So again, translate that into the opposite, your detecting rate instead of defect probability, and then you change that into the language of the detecting mRNAs. So you see, okay, we have a UMI count for this gene is 3, and how many mRNAs should be in there expressed, ready for you to try on, and until you get 3 read out. So I just basically change this to the language in single cell analysis. So there's multiple steps in the process of the detection, and there this reverse transcription rate, okay, amplification factor, dilution, sequencing dips. So if you really want to simulate those steps, and there's not only just negative binomial, and you probably will consider a lot of parameters. And some of those parameters are shared between cells because it's per run. But if you have another samples maybe run in different batches, then you probably have to adjust all this. So those are something which people will consider in order to understand the data and to infer what the real mRNA concentration is for every single genes in there. Once you understand those, you can start to regenerate and refill some zeros if you believe those are the missing values from the detection rate because of technology sensitivity is not high enough, so you miss that. So those are the technical dropout. People want to use different approach, try to impute those values. Okay, it's a 0. You believe it's supposed to be 0.05, so you reintroduce 0.05 into that particular location for a particular gene in particular cell. It's sometimes reasonable if you have a gene which all the cells, neighbor cells have a 15, okay, UMIs, and suddenly your self becomes 0, you probably want to fit that in 7.5 something, and because you know across the distribution it's supposed to be non-zero. But so people developed a lot of methods. I collect those. The top 6 is called model-based. So in those methods, we try to say, okay, there are some kind of distribution from the data. We want to regenerate them. There are other methods which not based on the assumption of any statistical model. It's just purely on the average cross data points, for example. So those like data driving and the smoothing method to take average. I don't care whether you generate it from a negative binomial or from a person or from a normal. I just fill the gap, interpret those data points, empty data points. There are deep learning method using neural network. So there probably need training data, okay? And you know how to set up those, preset those parameters in order to do a generated model, not based on distribution, but on some of nonlinear relationship and generate those. There are random factorization, maybe based on the multiplication of the matrix to do those. Anyway, so those are imputation method using statistical modeling. This is one example, and there are two distributions, and the one distribution gave the distribution of a parameter for another distribution. So another distribution will use this changed parameter to generate the data. So basically, there are two parameters for gamma distribution, create this mean every gene, a single gene in single cell, and then this lambda will be plugged in this Poisson distribution, and this Poisson distribution becomes the final data generators and pump up those values. So this will be 0, 1, 2, something like that, okay? But it's changed all the time based on another distribution of gamma to simulate the average cross cell population. Otherwise, if this is fixed, unchanged, all the cells have the same level of mean, so it's not realistic. Similar here, and this another SC impute method, do those steps, and also assume there's a negative binomial distribution then try to generate that, and compare with the real data, and do some iteration, and through compare with the real one, and start to adjust the parameters for negative binomial per gene, I believe. Then try to find combinations of the vector. Each element in the vector is expected average of that gene. Look at this and compare with the real data, and try to minimize the difference between your reconstruct and that original. At the end, to reach this purpose of imputation. Okay, we move to descriptive statistics, and now we are not talking about the distribution. We're not thinking about this underlying principle, the driver force for the data generation, but we are given a vector of data or a matrix. And then what we can do this understand the central tendency, which you use a mean or median or mode, and then the position and the rank. Okay, you can look at the frequency. So each bin, how many data points dropped in that bin, and then you describe the discrepancy, the variation. So what's the largest value and the smallest value? What's the range between them? For example, there's a standard deviation, those things. So based on those descriptions, you start to generate your hypothesis of the potential possible distributions, and start to estimate those values which can be used to regenerate your data. So this is a normal distribution with different parameters, average and variance, and give you different shape and the dispersion of this data distributed. And this is for PMF, okay, for discrete. And then you can add them up, and it becomes cumulative. And this is a density distribution. This is a cumulative. Cumulative is from 0 to 1. And you just add them up, all these bars. Okay. So when you move to the right end, the higher value, and you say, okay, when I move to 150 in this example, I will cover all the 100% of the data points. If I am in the 100 position, I will cover 50%. Then you look at the y-axis, you know the percentage, which data points you covered, basically. We use a lot of box plot, and this box plot was plot horizontally, but normally we have a vertical. And remember, this box was not just put there by good looking. There is meaning of that. In the middle, this bar is a median, and then you have a 25 percentile and a 75 percentile, so you know the points which distributed between them, okay? And then you have some decisions where you want to put outlier points. So there's some definition. Definitely people follow this rule. So once you see the box plot, you know, okay, how many points inside the box, how many outside, which data are how many are outliers. So this is a PDF, CDF, and the box plot for normal distribution. So you see the shape, the balanced, the percentile, okay? And this particular distribution have this plateau in the middle, and then you see the change of the CDF and gradually climb to 100%. It's not like steep as a normal distribution because of the plateau existing. And then also this bar will become wider. And in contrast, if you have a PDF, which is a sharp peak, and then you have very quick jump in your CDF. So this will, when you see this, you will have a picture on your mind is a distribution like that. And if you have a very narrow box, and then you know, okay, all the data points are very centralized in the mean or median. This is a skewed distribution, and then you have a long tail on the larger value side, so you keep going until very large x value you reach to 100% to cover all the data points, and this box plot distribution with outliers on your right side. If you have a bimodal distribution, then you have two peaks, and you start to see this quick increase in this part of this CDF, and then another increased place. Well, that information will be lost if you use box plot to show your data. So, okay, I think we can take 5 minutes break before time we should come back. Yeah, come back 11:01. Okay, so this graph is trying to explain what is a p-value. So if you have your assumption of the statistical model, so you know this distribution, so you know you have average at 4, and then the standard deviation like that, and give you this value. So based on this assumption, you can compute for any observed value. So if you observe a value which is located at this position here, and then you see, okay, based on my PDF, so here, okay, based on you, for example, CDF will be better. Then you see, okay, there are 95% of supposed to be observed data point will be smaller than this observed. Then you have your significant is 1 minus 95%. So this is just give you the idea. To calculate the p-value, when people say, okay, I got a p-value, your first question you can ask, as a universal question, is what's your distribution? What type of your distribution is? What your assumption is? Without answering that question, the people shouldn't know how they compute the p-values. A lot of people doing the permutation tests, they get the p-values, and you assume, oh, I have 0.01. What they did is try different random numbers and they say, okay, so they don't have assumption. So in that case, they should tell you we are using permutation tests instead of statistic tests. We have no model, it's model free, and get a p-value. They're not a standard p-value as a statistician will see, okay? So another small terminology concept is z-score, and this is called a standardized random variable. So you can get a vector of the data, say x, and you that's your observation. Then the x is mean, x bar is average. Then every value of your vector minus is mean, so you basically centralize your data to the 0, okay? So mean means half of the data have greater than this, maybe half smaller than that. So once you have your data point minus its mean average, then you get some positive value, some negative value balanced. Then that means you move your data points to the center, you centered by the mean. And then you divide by its standard deviation and put the variance of this value into 1. So once you do this transformation, it's called standardized, and your data becomes had this normal distribution around 0 and its variance equals to 1. So this is a very strong transformation to put every put the data point, no matter how far away from the 0, move to 0, and also no matter how dispersed they are, will push you back to with variance equals to 1. So when you use this transformation, you will be very careful, because you move your mean and you compare to another vector, and also you move to the mean, and then forces have a same distribution and also the variance. Okay, there's a very strong transformation. Standardize your variance. Another concept I want to mention is we also always have this problem called multiple testing correction. So people use so-called Bufferoni correction to give you Q value instead of a P value. Why we don't use this conventional P value like this? So you observe your data, you have your assumption, but why not use this nominal p-value? It's straightforward, 5%. Why you want to do this correction? The reason is you have your assumption like normal distribution, but you are doing not only one observation, but many. Just like you put this bin machine and you draw this bin into here many times. If you only draw once and your bin jump to this end, this is not a usual situation. This is unusual event. This is significant. However, if you do this hundreds, thousands of times, there's one time you bin jump to here. This is not so surprising because you are using multiple data points to rebuild your whole distribution. So you expect by doing this thousands of times, some of those observations should be far away from the center because you are rebuilding this whole distribution. So that's why when you see this observation within the multiple testing situation, you won't feel surprised because this is not a single event. It's not only one-time trial. It's continuous multiple trials. So that's why you need to set up even more stringent bar. You see, not surprising here, I have to set my p-value cutoff here instead of the conventional 5%. Okay, you want to set 0.05%, something like that, to correct your multiple testing problem. That's why people choose more stringent cutoff to do this correction. Instead of use p-value and you get a 0.05, I will divide by, you see the p-value cutoff 0.05. If you do n trials, say 1,000 trials, I will set my new cutoff p-value 0.05 divided by 1,000. So this is the most stringent cutoff. So I need to get a p-value smaller than this in order to claim it's significant after multiple correction. Okay, this is the purpose. So this is doing, and so your original cutoff is alpha, okay, 0.05. So if you only do multiple tests 5 times, you should set your cutoff 0.01. Using this to divide by that number. If you have 0.05, 100 trials, you should have a new setup to 0.005. This is called a Bufferoni corrected cutoff. And then if you have your p-values smaller than this, you will see, okay, it's significant. And you claim we have done this so-called Bufferoni correction, and my corrected p-value is still significant. In that case, you can claim you're okay. All these distributions are connected somehow, and one maybe another special case when some multiple parameter, one of them become fixed to 0, and then one distribution become another. So those are the potential links between different distributions. I will explain the Poisson distribution a little bit. So this is to simulate those discrete low frequent event. For example, in this case will be a traffic volume detection or estimation. So people probably know over the year and how many cars and pass this intersection. So if you have a large number here, divide by days and maybe 24 hours, 60. So you divide by this number and you get a p. This p really small, right? This means over the year, I have so many cars pass by this intersection, but now I calculate per minute on average how many cars I should have. This is like a average number of the cars, see, given lambda, okay? Average minute, maybe 3. So I calculate from a large number of statistics, and to calculate the frequency of a small event will happen. And that's my lambda 3, okay? Then I will compute, so what's the frequency if I stay here for 1 minute? How many cars will pass by? My probability of 2 cars will pass by is given by this bar, which is a little bit higher than 20%. What's the probability I observe this in next minute, there's 1 car pass by. This one is here, 15%. Okay, so that's this distribution means. So in order to create this histogram, we need one parameter is average frequency of this rare event, which is a lambda equals 3. If lambda equals to 7, for example, then this distribution will change the shape. It will move to the right side. So if you on average statistical for long term per minute, you expect 7 cars. Then when you stay here for another minute, you will expect maybe 4 cars or 5 cars, the highest possibility. Okay, you still have a chance, the 5% in this case, no cars pass by in that particular minute. So on this side, there are 3 curves. Each corresponds to different lambda. What we see before this lambda equals 3, right? Here, lambda equals 10, 4, 1. When you have 1, then 0 and 1 will be 37% equal. Then you have a average, we'll see 3, 2, then this one. So you know how to see this curve to observe, okay? Interpret. And that's the equation you compute how high each bar is. Basically, this p-value is how high this bar for given parameters, okay? So here they use mu instead of a lambda to calculate in the equation. Single cell will be the loading cell into droplets at a personal rate. For example, if you want to simulate this capture of the droplet captured cells, and then you see, okay, I know if I put 1 million droplet, and in my system, I probably will capture 10,000, and then I have this particular lambda values. And then you can start to compute in your real experiment how many droplet will capture 1 cell, how many will be empty, how many will be 2 cells in there. So you want to change those parameters, change this capture rate, and then maybe density of the cell, the concentration of preparation, in order to allow the capture of the single cell become maximized, right? So in this setup, you want to have this particular yellow curve, which higher probability to capture single cell rather than duplets and triplets, you still want to minimize the empty droplet. So some of those equations will be useful. And load bins into droplets. So another procedure, and also maybe rely on some of this personal rate. And also people start to use zero inflated model, and you can imagine you can have a zero inflated negative binomial model to simulate your data. This is like two steps of procedure. First, you will count how many zeros in your particular gene expression in the collected cells, and you set up a percentage, and then you model those zeros separate from non-zeros. And this is like a combined histogram. And on your left is a zero inflated model. You see this bar here, and the 40% of the cell of contains particular genes expression are zero, and then the rest of them using a different distribution to model, rather than just put everything into one distribution. Okay, so the two steps. I want to mention t-distribution with comparison between normal distribution. The reason we want to introduce this shape, because later we will do t-SNE, remember? t-SNE is commonly used dimension reduction technology. This T stands for t-distribution, so that's why we want to show this difference. This difference between these two PDF is the normal distribution is more centralized, and then t-distribution give you slightly more dispersed, and emphasize this right and left and right side, this thicker fit, okay, somehow like this. So if you generate, say, random number based on the standard normal distribution compared to t-distribution, you will find t-distribution assumption will give you more, slightly more data points in this region, okay, than normal distribution. So that region means the value which in the intermediate range, okay. So in t-SNE, people use that technology to replace normal distribution to allow those cells to be slightly away to each other rather than clustered in the center. So if you regenerate the cells in a plant form, then allow them to be away from each other a little bit, you will see them clearly, and they are not overlap onto each other compared to use a standard normal distribution. That's the purpose people use t-distribution, just push data points away from each other a little bit. Okay, so this is like a formula for compute mean and variance. And variance is you have your observation minus the expected mean and take square and then expect value for this difference from your points to its mean. Each PDF have its own analytical solution of what variance should be. So if you believe your data follow this distribution and you know those parameters, you have those set up already. Otherwise, you have to estimate from your data yourself. All right. By the way, those parameters also may be estimated from your data. To describe the central tendency, we use mean, median, mode. So those are the difference. Okay, to this. If you have a skewed distribution, otherwise they should be the same, centered. There are more descriptive statistics to describe the dispersion. So this is more important even for us. We want to use range, which is largest against the smallest value in your observation. You want to have a deviation, a difference between inch and the mean, variance, and the standard deviation. So on this side, you have, we will talk about the coefficient of variation, okay? So this we use a lot in single-cell data study, and this is called the coefficient of variation. This is standard deviation divided by the mean. What that means is you have a vector of data, then you calculate standard deviation, then normalize the standard deviation with the mean. And the reason doing this is these two parameters may be correlated. So for a data set with higher mean compared to another data set with another different mean, and then the data set with higher mean also have a higher standard deviation. So you want to control for that to make this variance comparable between these two sets of data, even though they have a different means. So that's the purpose of that. So you can also, so this is standard deviation. You can also rewrite this into square root of variance divided by mean. If you do that for Poisson distribution, then you have a variance equals to mean. So you can replace this variance, use mu. In that case, it becomes CV equals to square root of mean divided by mean. So that's an assumption for Poisson distribution. If that's the case, if you take a log between both sides, then you have a log CV equals negative 1/2 log mean. This become a linear relationship. That means if you compute for say genes by their mean and the CV, if you take a log of them, then you will have a straight line here. Okay, each point is a gene. You compute across all the cells. Okay, if those UMIs would generate using Poisson random distribution, follow that model, you will get straight line by looking at multiple genes. Higher mean, those genes should have a lower CV, and they are follow this straight line. And when we fit the real data, we can play with this in computer session, you will find a straight line. That means the real data follow Poisson distribution very nicely. No matter what 10x genomics chemical different version you use, you always get this straight line, basically. Very striking. So that's why statisticians really like single cell data, because they have a noise, they have zeros, but they follow their expectation very nicely. So they can say, okay, we also cannot predict very accurately what the next gene in that cell will be, the level should be, but collectively, they follow this beautifully. So we can do the imputation. We can see, okay, most of those technology generate data are follow distribution of the person. We know very well about that. Okay, so this illustration to show you how to calculate variance. All right, your data is in the fourth column of this X. You have 6 data points. Okay, this column X. So you compute mean first. So this X bar 14 is the mean. And then you compute each data point, Xi minus its mean. So in that case, you see sum of positive, sum negative. Okay, you put them together, should be 0. Then you square the value. Negative become positive as well. And then you sum them together. So this is the variance. This is the variance, and then this is this part. So we normally normalize by the sample size, because when you have more data points, then your variance become larger, and you want to divide by the number of n to take average. And the people found n minus 1 is better to give you less biased estimation from a theoretical expectation. So if you know those. So this is the way, the formula to calculate the variance. Okay. All right, not hard. So what the variance means? This gave you some idea how dispersed the data points you collected in that 1D space, basically. Okay, if you are highly dispersed, then you have a larger variance. Otherwise, you have more centralized distribution, then you have small variance. So this illustrates to calculate not the variance, but it's covariance that's between 2 vectors. Okay, so the variance is by itself. So within yourself, what's the difference, the dispersion. This is 2 vectors. 2 vectors, they are, see, 2 variables. Okay, in this case, we have samples, same number of samples. How many here? 9 samples. Okay, 2 variables, x and y. So you calculate, you centralize first variable first. Okay, by minor x bar and y minus y bar. So centralize those 2, then product, put together, multiplicate those values. Okay, you get this summation of total. If you replace y with x, you will get back the same of this as this. If y is same as x, so basically you compute variance with x itself. But this one is between 2 variables. And you get the value, and this value could be positive, could be negative. Depends on this value of summation of those products. So this YouTube illustrates another way to calculate covariance and the variance together, they use a very special this matrix format. At the end, you get this matrix with in the diagonal position the variance for different parameters, and the off diagonal position are the covariance between variables. Okay, so here variables are not translate into our single cell. It's if you study cells as a sample, then your variables will be the gene. Now, what this covariance matrix means is you look at this gene's expression across cells. If you look at this gene itself, then that will be the variance of this gene, whether they express high, low, whether there's very random or dispersed. The covariance means gene A and gene B, whether they co-evolved or co-expressed. And the positive basically indicate they're more likely to be co-expressed, and negative value will be the anti-correlation. Okay. Sometimes we plot out this scatter plot matrix, and those are the variables. And this each is a scatter plot for two different variables, and those are the same data points. Just look at each pairs of the variables and look at their scatter plot, and you can find the correlation between variables. Okay, so this will be like gene 1, gene 2, gene 3, gene 4, gene 5, gene 6, maybe 6 genes. And then you look at the gene 1 and gene 2 correlation, and each point should be a cell. Translated in that way. This 2 should be the same if you just flip it around, okay? Because x, y axis changed. This should be symmetric. So from the covariance matrix, you can get correlation matrix. We know correlation is positive 0.75 means, okay, some kind of significant correlation between these two variables. If you have a negative 0.8, there will be this slope of the line regression will be negative. The correlation matrix is somehow it's normalized covariance matrix. Okay, you get the covariance matrix and then divide by those values, basically. Values of the standard deviation of x and y. Any questions so far? Is it all right? Everything? Okay. Sometimes we want to compare and see whether your distribution is same as expected assumption. So here you have a normal distribution, then you do this so-called quanti, and then you look at your real data and you expected quanti for different values. And if you get a straight line, this so-called quanti QQ plot, that means that your real data distribution follow this, okay, this curve. You can also compare, say, two data sets. So this is from one data set, another from another data set. All you can do is you found a data points which is quantized at the same quantile level, and if you use two different data sets and found where this should be, if these two distributions are the same, you get a QQ plot, which in the all the data points project onto this 1 position, basically same quantile, same values. Okay, KL divergence. It's very important. The concept is based on the information theory. If you know the entropy or something like that, it also have a frequency and log the ratio. This inside the core here. There's an information theory behind that. So this is a measurement between similarity or difference between two distributions. In this example, you have a red curve compared to blue curve, and then you put basically you put a histogram the bins, okay, and to compare those bins frequency. Basically, this the height means frequency of those values. Okay. You draw it from inside that bin. For each bin, you compare that height from a 1 is like red against blue, and here red is almost high, and here is almost 0. So there's the ratio somewhere built in here. And within this overlap part, you have both. You have some values in there. Then you plug in this, and you calculate the KL divergence. And the KL divergence is not symmetric. And if you do KL divergence, A compare B and B compare A, you get different values. This is small examples. Instead of using this continuous distribution, using this 3 bar and this histogram to show you how to compute KL divergence. So your first distribution is like this. You have 3 values, 0, 1, 2. Your second distribution also is 0, 1, 2, but uniformly distributed. It's each 1/3 probability. But this one here is, this is high, almost 50% here in the middle one, and then really low for the value for the 2. So to compute that, you will have the distribution, this is P against the Q, and that's a percentage, okay, the height, the bar. And for the uniformly distributed, the Q distribution, you have the 1/3, 1/3, 1/3, will in this one, 0.36, and then 48, and 16. So you plug those numbers in this equation and take a log, take a ratio, and you get the KL divergence for comparison between distribution P against Q is 0.085. All right? This gave you estimation how different these two distributions are. KL divergence using Q as a reference against the P, then you get 0.097. Okay? So normally we just do divergence of P against Q and the Q against P, and take average. Then you get the two distributions KL divergence. That's Can you imagine a situation that we will use KL divergence in our analysis? For example, I assume my data was generated from negative binomial distribution, so I will set up these two parameters for normal negative binomial distribution. I will generate a lot of those data and make a distribution. Then I will compare with my real data. I assume real data also follow those distribution. And then I can use KL divergence to compare, say, the number I generated have a same or very different distribution as my observed. That place where we use KL divergence. Remember, it's not one number against another. It's the statistics, the histogram from one data set compared to another histogram from another data set. Okay? So the number you generated from the same gene, same cell, the first gene, first cell, may be very different from the first gene, first cell in observed data. But collectively, as a distribution, you expect, if you model that the data set nicely, use your statistical model, you should generate a very similar distribution. Then we use KL divergence to evaluate the success of your simulation. Yes?
So, is it better to use the KL compared to the QQ plot? Like, is it more?
Yeah, so QQ plot is like a visual detection. It's not give you the quantity. Yeah, QQ plot used a lot in the genetic study. You have a p-value which much higher within the sample compared to random. Yeah.
So you compare wild type versus mono?
It's not in that case. It's compare. Say you have 1,000 genes, and your assumption is 1,000 genes, none of them will be significant in terms of p-value. But you observed that you have say 50 of genes significantly higher. The p-value is smaller than some cutoff. So you can compare this to distribution of p-values using QQ plot. So in statistics, this is called the Simpson paradox. That is, if you look at these two, for example, genes expression, so one gene against another, if you look at all the cells, then you think when gene 1 express high and the gene 2 should also express high on average across all the cells. So you have a regression curve like this, go through. If you calculate correlation coefficient, you will get these two genes relationship in terms of the whole population of cells, a positive correlation. However, if you look at the two cells clusters and model the correlation, study the correlation between these two genes, one cell type after another, you'll see, okay, in both cases, they are negatively correlated. So this creates a paradox. See, in statistics, whether you will use a subpopulation of data to infer the relationship between variables, or you want to add more to get a global estimation of those. We don't know which one is correct. So that's a Simpson paradox. We have a lot of this situation. You use male mouse against female mouse. If you don't consider the sex difference, then you get one particular correlation between variables. But if you separate your animals into male, female, you get a completely different results. I think this last page we will see, okay, occasionally you probably heard about the likelihood, and they're related to the probability, but not the same. Not the same. The likelihood means that's how high you can get in your PDF, this distribution for any observed data points, which the probability or p-values sometimes means so how many the percentage or likelihood the data points will be smaller or greater than you observed. So you said you observed here, say 95 would be smaller than me, so I will be 5% significance. So that's the difference. And we use these terms to do this when you do optimization, and you see, okay, I want to follow, assume my data follow this particular distribution, but I don't know where the mean is. So I will try a lot until all the data points give me the summation of the likelihood and maximize my total likelihood. Then I know, okay, I should put my normal distribution mean in the middle of this data point. So what you are doing is to try to maximize your likelihood of all the data points. Okay, so if you move your curve into the middle of and you sum all this likelihood from all the data points based on your assumption, then you get the maximize the total likelihood. If you move away from that, a lot of those will give you small likelihood, so the summation will be so small. And also you can you use mean and the standard deviation, try to maximize the likelihood basically. Okay, so let me know any points which you have any confusion, questions. So you see there's no slides with a star on there, so this very general, go through everything, make you ready for next level. Okay, right.